This conference will now be recorded.
Good evening, students. I'm Dr. Divya. So now we are in the interesting topic of biology, right? So we are learning about genetics. So if you are understanding the best concept of this chapter, it is very easy for you to score NEET as well as board exam. And the thing is, you should be clear with these concepts. The main concepts should be clear for you. It's like how you're understanding the logic of this chapter or logic of genetics. It's very easy through all these courses and monohybrid course, dihybrid course, whatever we are going to discuss. Based on that, you can solve 80 to 90% of questions of this chapter. Okay. So if you're clear with these concepts, I'm sure that you will not lose any marks from this chapter. And the type of question come from this, any type question comes also, you can answer. So in this 5th chapter, principles of inheritance and variation, we are going to learn about, or we have already learned few concepts of this. We are learning about the factors which are controlling the character inheritance, right? So we were discussing about those things, factors which are controlling the character inheritance from parents to offsprings. So from parents to offsprings, characters are inherited through some kind of factors. So in genetics, it is the study of inheritance and variation. We have already discussed about the definition of genetics and all. So it's a branch of biology which deals with the study of inheritance and variation. So in this field, John Gregor Mendel was the first person who conducted a systematic experiment or a systematic study with the help of large data and observation of plants, right? He did crossing experiment in plants. Which plant he selected? Garden pea. In that he conducted his experiment with the large number of plants, large number of offsprings in the continuous generations. So if you are observing 1 or 2 plants, there may be a wrong conclusion, right? There is a chance of a wrong conclusion. But if we have large number of plants, many plants are repeating the same observation, we can conclude that that's how the characters are getting inherited from parents to offsprings. So that made him to conclude all these about the factors which are inherited from one generation to next generation. Okay, so he concluded his findings with the help of this experimental data and the analysis of this data. Up to that, people were conducting experiments, people were doing their studies in this field, but with the help of a supporting data, Mendel, the first person, he only conducted the first experiment with the support of a huge data. So Gregor Mendel is called as the father of genetics. So he conducted his studies in the garden pea plant. In which plant he did? Garden pea plant. And the scientific name of garden pea is Pisum sativum. Pisum sativum, that is the scientific name of garden pea. Why Mendel selected this garden pea for his research studies or research, why he selected that as a research material? What is the reason behind that selection? First of all, garden pea is a it's a short duration crop, right? So garden pea is having several contrasting characters which can be studied very easily. That is the main reason. Because if you are selecting a research material or plant material, that should have contrasting characters. Then only we can study that how one character is inherited from a parent to offspring. If the character which is not expressed in the proper form, how we can identify when it is passing through parent to offspring? That's why in this plant, Gregor Mendel found that there are several contrasting characters. Okay, there are several contrasting characters which are expressed in complete form, so that it is very easy to identify in parents and offspring, or what are all the changes between parents and offspring. That's why he selected, that was the first reason for the selection of this garden pea plant, and he selected 7 characters for his studies which can be easily studied. Then the second reason for garden pea plant selection is that it's a short duration crop. Okay, so he can cultivate very easily, and the cultivation is also very easy. If you are selecting a plant which is having 7 to 8 months duration or 1 year duration, how it is possible to conduct further studies? Because it will take from one generation to next generation. First of all, the crop maintenance throughout the year, it will be difficult. And not only that, from one generation to next generation, if it is taking so much time, it will be difficult, very difficult. So that to study the next generation, we need to wait 1 whole year. But in the case of this Pisum sativum or garden pea, the duration of the crop is very, very less. It's around 2 to 3 months. So from one generation to next generation, it will take very less time. So in each year, means in a year, they can study minimum 2 to 3 generations. So in this generation, they can make out the inheritance pattern also. Right? So due to all these reasons, Mendel selected Pisum sativum as the study material or garden pea as the research material. And he conducted plant hybridization. Plant hybridization, we know generally in plant hybridization, what and all we will do? First, we'll select a plant material. Here you can see the selection, the plant material. First, we need to select a plant material, and while selecting, we need to make sure that this parental selection, whatever we have made for crossing, those plants are called as parents, right? So those parents should be pure line. Pure line means if you are allowing to selfing, or again self-fertilization is allowed, it should form the same kind of individuals. There should not be any segregation of character. That means they should be homozygous. Parents should be pure lines or homozygous. Okay. So one male plant and one female plant will be selected for the study in usually in crossing experiment or in hybridization experiment. Then from this female plant, whichever plant we are selecting, from the female plant, from that flowers, when it is in bud stage, that time itself, the anthers will be removed. Okay, from the female plant, when it is in bud stage, that flowers are in bud stage, from that flower buds itself, anthers will be removed. Okay, and this is for what we are doing, that this is to prevent the self-fertilization or selfie. Okay, then next we'll cover those female plant. That again, why we are covering? To protect from the pollinating agents, to avoid unnecessary pollen grains from the other plants. Okay, next step, after protecting from the pollinating agents, next step is pollination. We need some kind of pollen grains in that for fertilization. So we have already selected the male plant which is having a desirable character. So from that pollen grains, we need only that pollen grains. Okay, then only we can make sure that the particular character, we are studying a particular character. If we are giving only that pollen grain, we can make sure that it is having that particular character. The next step is pollination. Pollen grains from that desirable male parent will be collected and dusted on the female plant. Okay. Then again, after dusting or after pollination, this step also we need to cover that plant. Why we are covering? Again, to protect from unnecessary pollen grains. Already there are one type of pollen grains, right? We need only that one type of pollen grain, and to avoid other pollen grains or unnecessary pollen grains, again after pollination we are covering that plant. Then allowing for fertilization. Fertilization, pollen grain and pollen pistil interaction that we have already studied. So again with that pollen pistil interaction, their fertilization will happen, and after that seed setting, right? So these crossed seeds can be used for raising further generation. With that material, we are going to raise further generation. So this kind of experiment Mendel conducted for 7 characters. Okay. So here, again, Mendel studied 7 contrasting characters. Okay. And he selected one character at a time for his studies. And this made that easy observation of characters and passage of characters through each generation. Okay. So he studied one character, only one character at a time. And if he is selecting more than one character, it will be confusing, right? So selecting one character at a time and observation of that character, which is passing from one generation to next generation. So like that, he selected 7 contrasting characters, and he made different crosses. For each character, he made different crosses. Okay. So there is no chance of a wrong observation. In one crossing pair, he was observing only a single character. And he selected parents which are having one contrasting characters. Here in this cross, you can see only one contrasting character. And for example, plant height he selected in this. And he crossed tall plant and a dwarf plant. Here you can see this is the tall plant and this is the dwarf plant he selected. Then here he crossed the plant and collected seeds after crossing. Okay, here you can see crossing work. Here you can see the crossing of tall plant and dwarf plant. Tall plant, which is having, this is homozygous tall and this is homozygous dwarf. There is no hybrid or a heterozygous condition. This is a homozygous, capital T and small t, right? That is in a homozygous condition or pure lines he selected for his crossing. And it produced gametes, and these gametes again in F1 generation. Here the parental generation. Next in F1 generation, you can see all plants were tall, right? That means F1 showed only one parental character. Okay, in F1 you can see all tall plants. That means that is expressing only one parental character. Then Mendel allowed these parents for selfing. Okay, here to produce next generation, he allowed these parents for selfing, and he produced F2 generation. So in F2, here after selfing, it produced gametes. Here you can see the gametes. These are the gametes which it is produced, and these gametes produced F2 generation. And in F2 generation, here you can see tall and dwarf plant. There are some tall plants. Here you can see this is tall, and the third one is also tall. But here you can see a dwarf plant. Okay. So, but there was no medium-sized plant in the F2 generation. Here you can see these 3 are tall, and the last one, this is dwarf. Okay. So, with huge data, so many number of plants he selected, it was there in his F2 generation, right? Here we are considering from a single plant. Like this number of plants were there in his observation, and he observed each and every plant and he made a conclusion. Okay. So there was no medium-sized plant. Here in F2, both the parental characters were expressed in Mendel's experiment. Here you can see, here one parent is tall, another parent is dwarf. In Mendel's experiment, in F2 generation, both the parental characters were expressed. But in F1 only tall parents were there. That means only one parental character was expressed. Okay. Here in F1, one parental character. But here in F2, both parental characters. Okay, that was there in F2. That means in F2 generation, here you can see 3/4 or 75% of plants were tall and 1/4. That means 25% of plants were dwarf. Both characters were there in F2. Both the parental characters were expressed in F2. In that, it was a 3:1 ratio. Here you can see out of 4 plants, 3 were tall and 1 was dwarf. Like that, throughout all the plants he considered, and among all the plants he made from that observation, he made this conclusion that expression of that parental characters, it was a 3:1 ratio. Okay, 3:1 ratio. But in that 3:1 ratio, we can see here the 3:1 ratio means it is the phenotypic ratio or the phenotypic expression. Here you can see different genotypes here. This is pure line or homozygous. Again, these two are heterozygous, but both will express the tall character only. Here dwarf is recessive. It is having both recessive alleles, so it is showing the dwarf character. Any one dominant allele is present means it will express only that dominant character, right? Then we learned about alphabetic symbolization in last class, as alphabetic symbolization of genes. And genes, we know they have different forms. A gene is having different forms. That different forms are called as alleles. Different forms of gene, it is called as allele. Then we used to mention one gene by using alphabet, right? How we are mentioning two different forms of gene for one character. See here, we are considering the character plant height. In that, how we are mentioning different forms? Capital T, capital T, that means tall. The dominant character we are expressing by using capital letter, and the same character recessive form we are expressing by using small letter. Okay. Here we have selected plant height as the character. That means tall. That is the dominant character. We are causing a tall plant, dominant plant with a dwarf plant. Here tall, how we will express with the help of alphabetical expression, how we will do? Capital T, capital T. This is homozygous tall, and homozygous dwarf, how we will mention? Small t, small t. And this will produce gametes. This will produce gametes, capital T, and it will produce only one type of gamete. And here homozygous recessive will produce only one type of gamete. Okay, that recessive gamete. Then in the first generation, how you will mention? In the first generation, you can see capital T, these, how this will form the combinations, these two and these two. Again, in the same way, here also another combination. So finally, you will get capital T, small t, capital T, small t, capital T, small t, capital T, small t. This will be the combination. Means in F1 generation, this is the parents. Okay. In F1 generation, all will form tall plants. Okay. You can see only tall plants. That is, what do you mean by this? There is only one parental character. Only one parental character. Here you can see the heterozygous condition. It's not in homozygous condition. Then also it is showing a parental character which is having the dominant character. Okay, the parent which is having dominant character. So tall condition is expressed here. Again, selfing. Selfing means capital T, capital T. Selfing means with the same pollen grain. So there won't be any difference. There is no other pollen grain. From outside, there is no other pollen grain. So here, how gametes will be produced? Two different forms of gametes will be produced from each plant, right? So how it will be? Here, you can see here. These both will combine, right? It will form capital T, capital T. And if this one is combining with this, it will form capital T, small t. And these both here, this combination, it is forming capital T, small t. And last combination, small t, right? So this, while writing this small t and this capital T, here one dominant allele is present. So first we will write that dominant allele. That shows dominant one will express. So here all these, here first while combining capital T, here it's wrong. Here see this. These two are combining and it's forming capital T, capital T. That one is dominant and means that will express tall condition. Again, capital T, small t. This combination also form a tall condition. Then third one, small t, capital T. Here also you will get this combination capital T, small t. That is also tall. Here small t, that is the recessive condition that forms dwarf plant. Okay. So there 3 tall plants and 1 dwarf plant. This is the phenotypic ratio or the expressed condition, right? Phenotypic means which we can observe. In that way, this is the ratio. 3:1 means 3 will be tall and 1 will be dark. But the genotypic or the genetic condition you can see here, it is genotypic ratio is 1:2:1. How that 1:1 means the first one expresses the homozygous tall condition. Next, 1:2:1. That is the ratio, genotypic ratio, right? Genotypic ratio. In this 2 is the heterozygous tall. Okay? And this is the homozygous dwarf. This is the condition. Got it? And next, then we learned about Punnett square, which is used to understand a typical monohybrid cross. So it was developed by Reginald C. Punnett. We learned about this Punnett square. It was developed by, who developed this? Reginald. Okay, who developed? Reginald C. Punnett. Okay. So Punnett square is the diagrammatic representation of a hybridization, then the production of gametes by the parents and the formation of zygote and F1 and F2 plants. What and all you can see in the Punnett square? Here you can see. Here you can see the parents and parental gamete formation from each parent. What and all gametes are formed. Then the formation of zygote. Here you can see the condition where zygotes, in which condition the zygotes will be. Then you can see F1 generation, then F2 generation. All this will be expressed in a diagrammatic way in Punnett square. Okay. So here you can see in a Punnett square the graphical representation. That is the graphical representation to calculate the probability or the chances or the probability of all possible genotypes of offsprings in a genetic cross. Okay. So in a genetic cross, what and all possible combinations will come, that we can find out from a Punnett square. So here in Punnett square, we used to write the possible gametes on two sides. Okay. On two sides. Here you can see the Punnett square, we are writing possible gametes on two sides. Here also we have given, and in this side also, and all possible combination in the boxes. Okay, that's the way we are expressing in Punnett square. And here, see, just now I told about tall. Here, tall plant and dwarf plant we are considering. Okay, so tall means capital T, and dwarf means small t, small t. So in Punnett square, we are going to write the gametes and possible combinations. Okay, in a simple way, we are going to mention all these things. So from this, which and all gametes will form? Capital T, capital T. And from this, which gametes will form? Small t, small t. Okay, these are gametes, so you should write in this way. Gametes will be expressed in this way. And here, the male parent will be, this is the male symbol and this is the female symbol. Okay. So here, this possible combination, capital T, small t, this is the possible combination. And here, again, this one, and from top, you can use this combination, capital T, small t, again. Again, this is the another combination, capital T, small t, and this is next, capital T, small t. This is the F1 generation combination. Okay, these are parents, and this is F1 generation combination. So in F1, again with the help of Punnett square, we are explaining the same concept. So just before writing, just read the question whether it is they are asking any monohybrid ratio with the help of symbols, or if it is with the help of a Punnett square, you have to check the question and write the answer. Okay. Here all tall. Okay. Again, in the second one, here all, like, see this, parent 1 is tall and parent 2 is dark, and it is producing possible gametes. From parent 1, capital T and capital T, these are the possible same type of gametes it's producing because it is homozygous. And here again dwarf parent is producing only one type of gamete, small t, and the possible combinations are capital T, here again capital T, small t, and capital T, small t, capital T, small t, capital T, small t. All are in heterozygous condition. So all are expressing tall condition in which one? In F1. All are tall. Okay. And when we are going to F2, in the F2 generation, here, from here F2 starts. In F2, what happens? F2 first, this is the, see, from here we are selecting one plant. Okay, that comes here, and it is selfing. F2 is not cross-pollination, it is the self-pollination. Self-pollination in the sense it is forming, it is crossing with the same pollen grain. Okay, there is no outside pollen grain. So what type of gametes it will produce? Again, we are forming the Punnett square. Here, how many gametes it will produce? It will produce capital T, one gamete, and another one will be like this, right? And from this, again, you will get same type of gamete, capital T and small t. See, while writing the Punnett square, there won't be any confusion in combinations, okay? You can, in this column, from left side gamete and from top side, another gamete. It is capital T, first combination. And in the second column, from top, this gamete, from left side, capital T gamete will come, and from top, small t gamete. This is the combination, another possible combination. Next, from here capital T and this gamete small t. This is another third possible combination. And in last column what we will write? Here, from here the small t, and again from here another parent, it is small t. So these are the possible combination in the F2 generation. Okay, F2 generation. So in F2, capital T, 1 capital T, then 2 capital T, small t, and 1 small t. Okay, 1 homozygous stall, 2 heterozygous stall, and 1 homozygous dot. That is the F2 condition. Okay. So in F2, the phenotypic ratio or the how plants are seen in F2. Tall and dwarf. That ratio we will tell. The phenotypic ratio is 3:1. 3 plants. Out of 4 plants, 3 will be tall and 1 will be dwarf. That means 75. In the total plants, total number of plants, 75% will be tall and 1 will be dwarf. Then the genotypic ratio is here. We express the genotypic, possible genotypic combinations we have already expressed in the Punnett square. Whichever is possible, everything we have written here. So that is 1:2, 2:1. In this, these are homozygous. This one and this one. One is homozygous tall and another one is homozygous dwarf. And these two are heterozygous, but it is tall because here one dominant allele is present, so it will express the character tall, but it is a heterozygous condition. Got it? So from the Punnett square, what and all we can learn? Punnett square, what is the condition or conclusion from the Punnett square? This Punnett square shows that the parental capital T, that is the male parent, and the dwarf small t, which is a female parent, these will produce the gametes. And then F1, what is the condition of that F1 genotype? It is capital T, small t, right? It is in heterozygous condition. Then F1 plants of the genotype. Here you can see this F1 plants of this genotype, they will undergo self-pollination. Then what they will do? And this F1 plant of the genotype, capital T, when it is getting self-pollinated, it will produce the gametes. Which type of gametes it will produce? Capital T and small t. Here you can see the possible gametes. And when fertilization is taking place, that pollen grain of genotype T, here you can see that this genotype T pollen grain, it is having 50% chance. This pollen grain from which is having the genotype T, it is having only 50% chance to pollinate. See, this condition I am explaining. Okay, this is having only 50% chance to combine with the same allele. Another 50% chance is to combine with the dwarf allele. So this one is combining the 50-50. There is a chance of 50-50. Here first it may combine with capital T. If it is combining with capital T or the same type of allele, that will form a homozygous condition. And the same thing if it is combining with a recessive allele, there is a heterozygous tall condition. Okay. So that's what we are explaining. Again, the same condition here also repeats. That's how it is forming homozygous tall and 2 heterozygous tall and 1 homozygous dwarf. Okay. There is a 50% chance for each combination. Next is about test cross, what we are going to learn. Okay. So by seeing simply by seeing a plant, we can't tell the genotypic combination, right? What is test cross? First, let me explain all these things. By seeing that plant, we can't make out what is the genotypic combination of a plant, right? We can say that either the plant is tall. See, here we are considering the plant height as a character. That's why always we are using this tall plant and dwarf plant, okay? Like this, Mendel selected 7 characters. So, by seeing a plant, we can tell that it is tall or dwarf. But dwarf means there is no doubt. Both alleles are recessive. Okay, this will be the condition, dwarf plant. But by seeing a tall plant, how we can say that whether it is homozygous tall or heterozygous tall? Okay, both are there, right? For both condition, it will express as tall plant only. So we can't tell that whether it is a homozygous tall. This homozygous condition also expresses tall. This heterozygous, here also one dominant allele is present, right? So it will express as tall only. It will never express as dwarf plant. This will form a tall plant only because there is a presence of dominant allele. Okay, so by seeing simply we can't make out the genotype of a plant. So, for example, whether here we are considering a tall plant from F1 or F2, whether it is having capital, here I said this condition, homozygous tall or heterozygous tall condition. Okay, so we can't predict the genotype of this plant. So therefore, to determine the genotype of a plant from F2, see in F2 you can see in F2 what is the ratio? 1:2:1 means capital homozygous tall, heterozygous tall, then homozygous dwarf, right? See, think about this plant. Okay, how by seeing it will be same like this homozygous plant. Both will express as tall. But we don't know what is the exact genotype. There are 2 chances are there. Either it will be homozygous dominant or heterozygous dominant. Both will be like tall plant only. So how to find out? So to determine this, the genotype, whether it is homozygous tall or heterozygous tall, for that we are using this test cross. So what Mendel did, Mendel crossed the tall plant from the F2. Okay, tall plant in the sense, see we need to find out the genotype of tall plant. Both are tall plant, right? We want to find out whether it is homozygous or heterozygous. That's the question. Okay. So what Mendel did, the plant which we want to find out the genotype, that plant he crossed with the recessive parent or with the dwarf plant. This cross is called as test cross. Okay. So the genotype unknown, that plant he crossed with the recessive parent. Recessive parent. I'll explain you how he did, then you can make out what is the use of this test cross. Okay. So, in a typical test cross, like here we are using pea plant. In that what he did, he crossed that genotype which is to be determined, which genotype is to be determined, that he crossed with the recessive parent. Okay. Recessive parent, he didn't allow that for self-crossing. Instead of that, he allowed for crossing with the recessive parent. And here we can observe, after this crossing again progenies will be formed, right? progenies or next generation will be formed. From the nature of this progenies, we can make out whether the organism is in homozygous or heterozygous condition. From progenies, from the appearance of that progenies, we can make out whether it is in a homozygous condition or in a heterozygous condition. Okay. So what we will do in this, the genotype in test cross, what Mendel did, that genotype, that unknown genotype, which is expressed as a dominant character. The genotype is unknown. The phenotype is expressed, but the genotype is unknown. That organism or that plant material or whatever may be, that one is crossed with the recessive parent, which is having the recessive character. So if we are crossing those two, progenies will be formed. With the nature, by seeing the phenotype of these progeny, we can make out whether it is a homozygous genotype, which we want to find out, or a heterozygous. Okay. So we'll see next how we can find out that. Okay. So here, let's see what we are going to do here. The same test cross we are going to explain with the help of Punnett square. So here comes the importance of Punnett square. We can easily make out, or we can easily identify the genotype. We can predict by using these combinations. Okay. So here the same test cross we are going to explain with the help of a Punnett square. The same test cross. Okay. Here we want to know the genotype of a plant, which is showing dominant phenotype, right? Here we are considering a tall plant. Okay, we are considering a tall plant. And that tall plant is having a dominant character, right? So we want to find out the genotype. What we need to do? We need to cross that with a recessive parent. So instead of self-crossing, we are crossing it with a recessive parent. That's a common procedure of a test cross. Then we are analyzing the progenies of a cross, and what we will do with that, we can determine the genotype of this tested organism. So here we are considering another character. The test cross here we are doing with violet color flower. You can see here violet color flower, and this is the violet color flower, and this is the white color flower. Okay. In this violet color flower, violet color is dominant. Okay. And white color is recessive. Got it? Violet color, we are using the symbol capital W. This is dominant. And next, white. Another type of flower is white. This is with small w. This is recessive. Okay. So now we'll try to find out the nature of offsprings of a test cross, or we'll check which ratio we will get. Okay. So the first flower, that is homozygous recessive. Here you can see homozygous recessive. That is in recessive means small. This is a white flower. Okay. Then here look at this violet flower. So we need to find out the genotype of this violet flower. So which is showing a dominant character. Violet, we have already told violet is the dominant character and white is the recessive character here. So which is showing a dominant character in violet color. Here, see this flower, this cross. Here we are going to cross these two flowers. Okay. This is the recessive parent in homozygous condition. So in a test cross, what we will do? Here the genotype of this we need to find out. There are two possible combinations. Like if it is violet color is the dominant color, and this genotype we want to find out. We want to know the genotype of this violet color flower. So If it is a dominant allele, if both the alleles are dominant, then also it will show violet flower. And if any one allele is dominant, when one allele for the violet color it is having, then also it will show a violet color. So these are the possible combinations. And now we need to fix which one is this. Okay, which is the possible genotype. These are two possible combinations, two possible genotypes of violet color flower. So we need to determine which one we are having. Okay. So the unknown genotype, either it may be capital W, or capital W, or small w. Okay. So here dominant genotype for this genotype is unknown, right? So we have two chances. Which are all the two chances? Either it may be capital W, homozygous dominant condition. This forms violet, or here both alleles are same, both dominant alleles are present, or it may be in heterozygous condition, capital W and small w. Here heterozygous, both alleles are different. Here capital, one allele is capital W, another one is small w. That means in this condition also it will form violet color only. Right? So let's explain with the help of a test cross. Here, see the first chance. First chance is both dominant alleles. Both alleles are dominant. Okay. So in the first chance, how it will be? In the first chance, see here. 1 second. We'll write both possible combinations here. Okay. So here, which are the possible combinations? One parent is recessive. That is confirmed. This is the white color flower. Right. And we are crossing this with violet color flower. Violet color, first possible chance is both dominant alleles. Okay, for violet, first chance. So this will produce, here with the help of Punnett square, we are going to explain again. With the checkerboard, we are going to explain. This will produce two alleles like this, both are similar, and this also again you will get capital W, these are the gametes, you can see. And in this, capital W, small w, next combination, this one and this one, capital W, small w, another combination, capital W, small w from this and this. From this, you can see capital W and small w. Okay, so here, all are violet flowers. All violet. See, if your genotype is a homozygous dominant, all are violet. You can see all violet flowers. Okay. So if to find out the genotype, if all are showing similar type of flower or violet color flower, your genotype is the homozygous dominant. Okay. So one confusion got over. Next is, which is the next condition? Here, white color flower is fixed. We are going to cross with the recessive one, right? So next chance is, which is the next chance? Here, this one, heterozygous condition, heterozygous dominant condition. This is the heterozygous dominant condition. This is a violet flower, right? We want to know the genotype of this, whether it is a dominant homozygous or dominant heterozygous. That's what. So we are taking the second combination. In this also, again, this recessive parent will produce same gametes, and here there is a difference, right? One dominant allele and one recessive allele it will produce. So the first is capital W, small w, then second combination again capital W, small w, third combination is small w, and here again the combination is small w, small w. Okay, so here half of the flowers are violet. Okay, and another half, next half is white. Okay, half of the flowers are violet and next half is white. So what conclusion we can make from this? If the dominant phenotype or the violet flowers, by seeing the offspring of the test crows, see we need to see the offsprings of these test crows. Then only we can predict the genotype of this tested organism. Okay. So if that testing genotype is homozygous dominant, all individuals or all offsprings formed will be having dominant character. All will be having dominant character. If it is homozygous dominant, here you can see if it is homozygous dominant, all will be having dominant character. All the offsprings will be having dominant character. And if the tested genotype, if this tested genotype is having a heterozygous genetic condition, half of the offsprings will be having one parental character, that means dominant character. Another half will be having a recessive character. Here you can see half will be violet and half is this half of the offsprings. They are violet and another half is white, right? So it will be the distribution of offsprings will be, or offspring character of the offsprings will be in 1:1 ratio. 50% of the offsprings will be showing dominant character and another 50 will be showing recessive character. Got it? So that is the test cross. So what is the definition of a test cross? In a typical test cross, an organism showing a dominant phenotype. If it is showing a dominant phenotype, it is crossed with the recessive parent, and it is crossing with the recessive parent instead of self-crossing. The progenies of such cross can be easily analyzed to predict the genotype of the tested organism, right? So how we can find out the genotype? It should be crossed with the recessive parent, and by seeing the offsprings, we can predict the genotype of that tested organism. If the genotype is homozygous dominant, how the offsprings will be? All the offsprings will show dominant character. Okay. If the tested organism is heterozygous, all offsprings, how we can see? 50% will be dominant and another 50% with recessive character. So that is the conclusion Mendel made. Okay. Half will be dominant and half will be recessive if the organism is in heterozygous condition. Okay. If it is in homozygous condition, all the offsprings will show dominant character only. Next, based on all these observations on monohybrid process, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Okay, so based on all these experiments, Mendel proposed two laws which are called as principles or laws of inheritance. Principles of inheritance or laws of inheritance. Mendel proposed Principles of inheritance. Okay. Or principles of inheritance or laws of inheritance. And first law is regarding the law of dominance, and second law, it is regarding the law of segregation. First law explains how a character shows dominance, and second law, this principle of inheritance, in that second law shows how one character is getting segregated. Okay, that is regarding the segregation of characters, and first law is regarding the dominance of one character. Okay, like that Mendel proposed principles of inheritance or laws of inheritance by understanding the offsprings of a monohybrid cross. Okay. So in first law of dominance, what it says, law of dominance? In law of dominance, characters are controlled by discrete units called factors. Okay, this law of dominance you need to study properly. The characters are controlled by factors. Mendel called the genes as factors that time. Okay, and these factors occur in pair. Okay, we learned that. Now we know the tall character, they are controlled by genes or factors. Mendel called as factors, genes or factors. Got it? And these factors occur in pair. We learned that there are different forms for genes that are called as alleles. Now we are calling it as alleles. So factors occur in pair. Okay. That's why we are writing these two alleles for a character. And in a dissimilar pair of factors, one member of the pair dominates and the other one, that is the recessive one. Okay. In a dissimilar pair, here this is similar pair. In a dissimilar pair, that means in the heterozygous condition, here two alleles are present. But here the dominant one will express. That's what it says. The dominant one will be expressed and the other one, dominant one, this expressing one is called as dominant allele, and the suppressed one, that is called as a recessive allele. And the law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross. Okay. And in the F1 and the expression of both in the F2. Okay. So in a monohybrid cross, it will explain in F1, it will explain only one parental character. One parental character will be expressed according to this law of dominance in a monohybrid cross. In F1, only one parental character will be expressed, and in F2, both the characters will be expressed. So it also explains the proportion of the F2 phenotype, 3:1. In that ratio, in F2, organisms will appear in 3:1 ratio. That is the law of dominance. So law of dominance says that all characters are controlled by factors, and these factors, or nowadays we are calling these factors as genes. So these factors or genes occur in pair, and in a dissimilar pair, means in a heterozygous condition, one dominates over the other, and the dominant or the expressed one is called as dominant allele, and the other one, the suppressed one, is called as recessive allele. And according to the law of dominance in a monohybrid cross, in F1, one parental character will be expressed, and F2, both the parental characters will be expressed. And the phenotypic ratio or the expression will be in the ratio of 3:1. Okay. Next is law of segregation. What this law of segregation says? In law of segregation, this is based on the fact that alleles do not show any blending. See, we have already explained in F2, either it will be tall parent or it will be a dwarf plant. Okay, there won't be any blending because in F2 generation, both the parental, either tall character or dwarf character will be expressed. There is no medium sized plant. There is no in between character. Okay. So both the characters are recovered as such in the F2 generation. Right. In F1 only one character is expressed, but in F2 generation both the characters are recovered. Though one of these is not seen in F1. In F1, one parental character will not be expressed, but in F2 both the characters will be expressed. Okay. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Okay. So, even though in heterozygous condition, see, we have already told in heterozygous condition, while gamete formation, it will form two types of gametes, right? And there is a 50-50 combination, chances for combination. Okay. And of course, a homozygous parent produces all gametes that are similar. A homozygous plant will produce similar gametes, while a heterozygous one will produce two kinds of gametes, each having one allele with equal proportion. So this is also, this dominant allele is having 50% chance, and recessive allele is also having a 50% chance of combination. That's what law of segregation says. Got it? Law of segregation. There is, in heterozygous condition, one parent produces two kinds of gametes, and each are having 50% of possible combination. Okay. That says the law of segregation. Here in F1 and F2, how we will say about this law of dominance and law of segregation? See, with this example, again, tall, homozygous tall and homozygous dwarf. With this, how gametes are produced? Capital T, we are crossing this plan, capital T, capital T. And here, small t, small t. Okay. When it comes to F1 generation, how we will say capital T, small t, capital T, small t, again, capital T, small t, capital T, small t. Okay, this is F1. So here law of dominance, see here dominant allele is present. So that dominant allele will be expressed. That's why all are tall. This is, here comes the law of dominance. Okay, law of dominance. Again, this is a symbol for selfing. Okay, so this plant is selfing. Capital T, capital T, capital T, capital T. Right, this is the selfing. It crossing with the same thing only. So here it produces capital T, small t, capital T, small t. Here segregation, both the parental characters are expressed. How we will get? One capital dominant, homozygous dominant, two heterozygous dominant, and one homozygous recessive we will get usually. Right. So in this, both the characters are expressed in F2. This is the F2. So here comes law of segregation. So in law of segregation, both the characters are expressed. Here you can see in F2, both the characters are expressed. Here tall, one parent is homozygous tall, and both type of gametes are produced in F2 generation, right? And here, this is also tall. Here, this is dwarf. That means both the characters, both the parental characters are expressed in F2. So that's how here we are applying law of dominance and law of segregation in each process. So that's all about Mendel's law of inheritance. He proposed from a monohybrid cross law of dominance and law of segregation. So hope it's clear about the test cross and all. So go through these portions. Next class we will be learning about the further aspects of genetics. Thank you.